Hello, welcome back. So we are saying here that at each stage or time during the cell cycle, each of your chromosomes is associated with a group of proteins called histones. So you can see this association here. So DNA wraps around these histones at regular intervals to form things similar to beads on a string. So parts of the histones interact with each other, further compacting the DNA. And at this stage, the spaghetti stage, or the loose combination of DNA and protein, is called chromatin. So this compacted thing is what we are calling chromatin. So the word actually means loose. Describe how much the DNA strand folds back on itself. It does not mean that the DNA is loosely wrapped around the histones anyway. So as you can see, they are not loosely wrapped, though the word may mean so. So as a cell progresses into mitosis, chromatin further condenses. It continues to coil more and more tightly around organizing proteins, finally forming small thick rods. So recall that each chromosome that has already been copied during the previous stage, that is the S stage or the synthesis stage. Thus, the chromosome looks similar to an X. So the chromosomes will be looking similar to an X, as you can see here in the diagram, in which the left and the right half side are two identical double helix structures, are two DNA double helixes. So one half of the duplicated chromosome one half of the duplicated chromosome is called chromatid. So this half here is a chromatid. So together, the two identical chromatids are called sister chromatids. So this is one chromatid and another one. So we have here chromatid, and the chromatid is held here. So we have one chromatid, you have another one, and they are held here at the center by centromere. So in addition to the end of the DNA, molecules form structures called telomeres, which are made of repeating nucleotides that do not form genes. So these telomeres do not form genes, do not form genes. This is very important for the cell. So this prevents the ends of the chromosomes from accidentally attaching to each other, and they help prevent the loss of gene. So this also helps prevent the loss of genes. So a short section of the nucleotides, I mean a short section of the nucleotide is lost from a new DNA molecule each time it is copied. So each time DNA is copied, there will be a loss of short nucleotides. So it is important that these nucleotides are not lost from, are lost from the telomeres here, and not where we have genes. So if it is lost in the telomeres, it's no big deal. But if it's lost in the cells, it will be a big deal because it will be actually loss of a gene is a big problem. It will be bring about a mess. So welcome back. So we are saying here, mitosis and cytokinesis produce two genetically identical DORA cells. Produces two identical DORA cells. So what we mean here by DORA cells being identical is that the DNA is actually copied and also the other organelles inside the cells are doubled and divided into two genetically identical cells. In fact, the main function of mitosis is to produce cells for growth or repair. And in rare cases, also in reproduction, as in terms of asexual reproduction. So the combined process of mitosis and cytokinesis produce two genetically identical cells. The combined processes of mitosis and cytokinesis and cytokinesis produce two generically identical produce two generically identical DORA cells. So during interphase, which is the first part of mitosis, so we are having a cell like this one here. Sorry. So this is the cell we are having here. So before the cell divides, we are having a single nucleus. So before it divides, actually the DNA has to be replicated. Then the nuclear envelope has to break down. So what we have here are centromeres, or I mean centrioles. Then we have spindle fibers which will develop across the cell. Then we have centrosomes. So the first part of mitosis is, or I mean cell division, is interphase. During interphase, you know what happens. It plays an important role in preparing the cell to divide. So interphase plays an important role in preparing the cell to divide. Plays an important role in preparing a cell to divide. That is what it does best. It prepares the cell to divide. It provides critical time for duplication of organelles. It provides critical time for cell organelles for the duplication, for the duplication of organelles. So these organelles have to be duplicated, and for DNA replication. Because we said at this time here we run from here up to here, maybe we have the S phase or the synthesis. This is the G1, then we have the G2. So we are meaning that it provides critical time for the cell to duplicate and for DNA to replicate. So by the end of interphase, an individual cell has two sets, full sets of DNA or chromosomes, and is large enough to divide. At the end, the cell, by the end of interphase, the end of interphase, the cell has two full sets of DNA. So it will be having two full sets of DNA or chromosomes to be precise, and a large enough and is large enough to divide. So it would be ready for division. It would be ready to divide at this time. Then now it will go to the next stage, which is mitosis. So as you can see, mitosis here is highlighted in red. That is mitosis. So mitosis divides a cell nucleus into two genetically identical nuclei. That is what mitosis does. Mitosis divides a cell's nucleus into two generically identical nuclei. Into two generically identical nuclei. Each with its own single full set of DNA. Each with its own single full set of DNA, each with its own single full set of DNA. So this process occurs in all your body cells except those that form eggs or sperms. Of course, those ones, the gametes. So for them, they will not go through mitosis, but through another type of cell division referred to as meiosis. So this process occurs in all body cells, as I have said. So although mitosis and cytokinesis are continuous processes, so these two here, this is cytokinesis, this is mitosis. Let me write it here. Mitosis, cytokinesis. So even though they are actually continuous, scientists have divided them into phases to make them easy to understand and discuss. So the four main phases of mitosis are P, M, A, T. So it's easy for you to keep it in your mind. P mat. So P for profess, M for metaphase, A for anaphase, and D for telophase. So these are the four main divisions. So we will discuss them into details, and we will do this by, of course, the last part of cell division is cytokinesis, which normally occurs after mitosis. So we will look at what happens during this cycle, what really happens here. So figure 2.4, the cell cycle in details. So following interphase, mitosis divides duplicated chromosomes between two nuclei. Cytokinesis divides the cytoplasm. So we will be looking at them in the next video, and we will discuss it all.